Hey everyone, welcome back to MLE Masters, your free video series to help simplify complex medical topics and make high-yield learning effortless. Today we're diving into mitosis, a fundamental process in cell division that's not only crucial to life, but also heavily tested in exams like USMLE Step 1. We'll walk through each phase of mitosis in a clear and simple way, touch on relevant pharmacology, and show you how this topic connects directly to clinical medicine. Mitosis is the process by which somatic cells, that is, all the cells in our body except our sex cells, divide and form two identical daughter cells. These daughter cells are genetically identical to each other and to the parent cell, and they are diploid, meaning they carry two complete sets of chromosomes. This process is essential for replacing damaged cells, for normal tissue growth, and for maintaining the integrity of our organs and systems. So yes, mitosis is important for life itself, but for medical students, it's even more important because every phase of mitosis can show up on your exams, often with pharmacology tied in. Before we enter mitosis, the cell goes through a preparatory phase called interphase. Interphase includes the G1, S, and G2 phases. During the S phase, the cell replicates its DNA. In G2, the cell grows and prepares for mitosis. Although interphase is not part of mitosis, it lays the foundation for everything that follows. Now let's go through the phases of mitosis, starting with prophase. In prophase, the DNA that was previously in the form of loose chromatin begins to condense into visible chromosomes. The nucleolus disappears, and spindle fibers made of microtubules start forming from the centrosomes. These spindle fibers begin to connect to specific parts of the chromosomes called kinetochores, which are located at the centromeres. The centrosomes move to opposite poles of the cell, forming structures known as asters. Some sources include a separate stage called prometaphase, where the nuclear envelope breaks down completely and the spindle fibers firmly attach to kinetochores. You can remember this as a transitional step between prophase and metaphase, but for exam purposes, it's often considered an extension of prophase. Next comes metaphase, a highly important checkpoint phase. In metaphase, all the chromosomes align along the metaphase plate, right at the center of the cell. This alignment ensures that each daughter cell will receive an exact copy of genetic material. The cell checks to ensure that each sister chromatid is properly attached to spindle fibers at its kinetochore. This is called the spindle checkpoint, and it prevents errors in chromosome segregation. Once all chromosomes are properly attached, we move into anaphase. In anaphase, the sister chromatids are pulled apart and moved to opposite ends of the cell. The spindle fibers attached to the kinetochores begin to shorten, dragging the chromatids toward the poles. At the same time, other spindle fibers that are not attached to chromosomes elongate, pushing the cell poles further apart. This coordinated push and pull ensures that each new cell will have a complete set of chromosomes. An important motor protein called dynin plays a role in this movement. Dynin attaches to kinetochores and walks toward the negative end of the microtubule. You can remember this because dynin sounds like die, and death is negative, so it moves toward the negative end. Following this, we enter telophase, the final phase of mitosis. In telophase, chromosomes begin to decondense back into chromatin. The nuclear envelope reforms around each set of chromosomes at both poles, and the nucleolus reappears. The mitotic spindle breaks down as it is no longer needed. Simultaneously, cytokinesis. Cytokinesis is when the cytoplasm divides, and the cell pinches in the middle to form two identical daughter cells. Each of these cells is diploid and genetically identical to the original parent cell. To remember the stages of mitosis in order, you can use the classic mnemonic: I passed my anatomy test, interphase, prophase, metaphase, anaphase, and telophase. Now let's talk about some high-yield pharmacology. Several drugs used in medicine specifically target the mitotic process. Let's review three key drug classes. First, colchicine. Colchicine is commonly used to treat acute gout. It works by inhibiting tubulin polymerization, preventing microtubules from forming. As a result, spindle fibers can't form, and the cell becomes arrested in metaphase. It also inhibits neutrophil chemotaxis, another reason it's effective in gout. Next, Vinca Alkaloids, drugs like Vincristine and Vinblastine. These also inhibit tubulin polymerization and halt mitosis at metaphase. Vincristine is commonly associated with peripheral neuropathy, while Vinblastine tends to cause bone marrow suppression and is used to treat Hodgkin's lymphoma. Then we have taxanes, such as Paclitaxel. Unlike colchicine and Vinca Alkaloids, taxanes inhibit tubulin depolymerization, preventing the breakdown of microtubules. This mainly affects anaphase, since depolymerization is needed to pull chromatids apart. Side effects of taxanes include neuropathy, anemia, hair loss, thrombocytopenia, and other common chemotherapy symptoms. Let's bring this home with a clinical case. A 45-year-old man presents with severe pain in his right great toe. No trauma, but warmth and swelling. Classic gout. The first-line treatment is colchicine, and now you know it acts during metaphase and mitosis. That's how clinical medicine and cell biology connect in real board-style questions. And that brings us to the end of this session. I hope this video helped you understand mitosis, its phases, and the pharmacology tied to it. If you found this helpful, don't forget to like, share, and subscribe to MLE Masters. We're here to simplify learning, one topic at a time. Thanks for watching, and I'll see you in the next video.